Hello friends, welcome to the channel, and today's video is about Krebs cycle, the second important pathway in carbohydrate metabolism. So now let's see what is Krebs cycle and how it starts with. So in the previous lecture we have already seen that glucose get converted into pyruvate through a 10-step process, that is glycolysis pathway. Now this pyruvate, it enters into mitochondrial matrix, and here it gets converted into acetyl CoA. So I have shown here acetyl CoA with this two circles and a box. So this is denoting coenzyme A, and these are the two atoms. Means acetyl coenzyme A is a 2-carbon molecule, and it is attached with a CoA. Now this step is catalyzed by an enzyme which is called as PDH. That is pyruvate dehydrogenase complex enzyme. Now when there is dehydrogenase, always remember there is utilization of NAD+, which leads to generation of NADH. It leads to formation of NADH. This is an example of oxidative decarboxylation, and here in this process one CO2 is released. So this is the conversion of pyruvate to acetyl coenzyme A, and this acetyl coenzyme A is the starting material for initiation of the cyclic pathway, that is Krebs cycle. Now why we call Krebs cycle a Krebs cycle? Because it was invented by Professor Hans Adolf Krebs. So in his honor it is named as Krebs cycle. It is also called as citric acid cycle, or it is also called as tricarboxylic acid cycle, or TCA cycle. So those are the other names. So now what happens with this acetyl coenzyme A? So we will be seeing only the substrates, and we will learn to recall their names in this pathway in this video. So what happens with this acetyl coenzyme A, which is a 2-carbon containing molecule, it reacts with, or it gets fused with the other molecule, that is oxaloacetate, OAA. I am writing the short form OAA, oxaloacetate. It is having 1, 2, 3 and 4. It is having 4 atoms. So these 4 atoms and these 2 atoms refuse together, and that leads to formation of a new molecule which is having 1, 2, 3, 4, 5 and 6 atoms, and it is named as citrate. So in this way, citrate is produced by the fusion of acetyl coenzyme A with oxaloacetate. Now this citrate undergoes isomerization to produce the next molecule, which is again having 6 atoms, that is 1, 2, 3, 4, 5, 6, and that is called as isocitrate. And this is a simple isomerization step. So we got the second substrate for Krebs cycle. Now we have to remember the next substrate. So there is a simple mnemonic for that. Just learn, just concentrate to understand it. Citrate is Krebs starting substrate for making oxaloacetate. So what is it? Citrate is Krebs starting substrate for making oxaloacetate. So these are the substrates mnemonic, and how we will correlate it with the name of substrates. Now let's see that one by one. Now we have seen conversion of citrate to isocitrate by a simple isomerization step. In the next step, this isocitrate, which is having 6 carbon, it gets converted to a molecule having 5 atoms, that is 1, 2, 3, 4, 5, and that is called as alpha Kg, that is alpha ketoglutarate, which is having 5 carbons. So it means there is loss of 1 carbon in between this step, and generally this loss of carbon is in the form of carbon dioxide, CO2 will be released, and this is simple decarboxylation process. So this decarboxylation, isocitrate got converted into alpha ketoglutarate. So that is Krebs alpha ketoglutarate. Then in the next step, this alpha Kg gets converted into the next material that is starting. So that is for succinyl CoA, succinyl coenzyme A. And again you could see here this is 5 carbon and there are 4 carbons in the next substrate. So it means there is loss of 1 carbon. So that loss of 1 carbon is with decarboxylation, there is removal of 1 carbon dioxide molecule. And here a coenzyme A is attached. So there is incorporation of CoA during this step. Then this succinyl coenzyme A gets converted into the next substrate, and now this substrate is succinate. Name of next substrate that is starting with S, so that is succinate. Now this succinate further gets converted into F for, so that is fumarate. Fumarate reacts with some water and it makes malate, M-A-L-A-T-E, fumarate to malate. And this malate then gets converted into oxaloacetate and the cycle continues. Then this molecule of oxaloacetate combines with the next molecule of acetyl coenzyme A, which we got from the second pyruvate, and again there will be formation of citrate, isocitrate, alpha ketoglutarate, succinyl coenzyme A, succinate, fumarate, malate and oxaloacetate. So in this way the cycle will continue, and it for one molecule of glucose this cycle is repeated twice for complete metabolism of a glucose molecule. Now the next important thing that I want to discuss here is about that the Krebs cycle is a central metabolic pathway. And why we call it so? Because all the substrates which we have seen here, I just want to connect you along with the substrate and the other metabolic pathways. So what happens in this step, why we call it as a central metabolic pathway? Because the substrates which are produced here like citrate, alpha ketoglutarate, succinate, fumarates, malate, oxaloacetate, succinyl coenzyme, they act as substrate and for the synthesis of other biomolecules like fats, non-essential amino acids, for the synthesis of glucose like oxaloacetate and malate, they get converted into pyruvate and then the gluconeogenesis is initiated. Similarly, citrate and they all help in the synthesis of even purines, pyrimidines, fatty acids, non-essential amino acids. So this is about Krebs cycle, the central metabolic pathway. Thanks for joining and keep watching. Thank you.